Now we'll be studying about Human Genome Project. We have already studied that it is the sequence of base which determine the genetic information of given organism. Or you can say genetic makeup of an organism or individual lies in the DNA sequence. Okay, and if two individual we find differ, it means their DNA sequence might be different, at least some places. And these assumptions led to the query for finding out the complete DNA sequence of human genome. Genome means haploid set of chromosomes. Now, with the establishment of genetic engineering technique, where you are able to isolate the DNA, you are able to clone any piece of DNA, and it is very simple and it is fast technique. So obviously, we were thinking to know the sequence of the DNA, and we say it is a very ambitious project which was launched in year 1990. It was called as mega project. Why we call it as mega project? Because if you see the time, money, so much requirement is there. First of all, we are having this many base pair. In the genome, this many base pair are present. And if we have to do the sequencing of one base pair, it is going this much. Means $3 per base pair. It means what is the cost of the total human genome sequencing? The total human genome sequencing means for the sequencing of this much base pair, total cost will go approximately $9 billion US. The money requirement is very high. Second is if we will try to store this information in a book, and if each page of the book contains 1,000 letter, each page contains 1,000 letter, and each book contains 1,000 pages, then we want 3,300 books to store the information. So it means lot of data will be generated. So as a result, we want high-speed computer to store this data, to retrieval whenever we want, to analyze it also. So we can say Human Genome Project was associated with the rapid development of new area that is known as bioinformatics. Now, what was the goals they made? First goal was to know the sequence of 3 billion chemical base pair. Second is to identify 20 to 25,000 genes approximately which is present according to them. Third, store this information in the database. You have to improve the tool for data analysis. We have to transfer the information to other sectors like pharmaceutical companies or food industries. And after this, we have to address the ethical issues because few people will say, why you're doing the disturbance with the God creation? No need to do this. Ethical issue, legal issue, or social issue, which may arise because of this project. So it was started in 1990 and they were targeting, we will complete within 13 years. So Human Genome Project was 13-year project because it is started, if you check, it is started in 1990. Okay, check carefully. The project was launched in year 1990 and they completed this project by 2003. It means total year is, it is 13-year project. 13-year project, which was first started by US Department of Energy and National Institute of Health. Later on, another contribution came from the Wellcome Trust of UK, okay, and other contribution, major contribution, additional, you can say, additional contribution came from Japan, France, Germany, China, and others. So started by US Department of Energy, National Institute of Health. Later on, Wellcome Trust of UK became major partner and additional contribution came from Germany, Japan, France, and China also. So knowledge about the effect of DNA variation, what will be the role because of the DNA variation, and you can say it is actually it can lead to revolutionary new way to diagnose any diseases, to treat the diseases, and someday might come where we can prevent the thousands of disorders which affect the human being. So it is not only providing the sequencing is not only providing the clue how to know the human biology means how to know the sequencing in the human base pair. Learning about non-human organisms has also helped us to know how these other organisms are able to survive in their natural capabilities so that we can solve the challenges which we face in the healthcare or in agriculture or energy production or environmental remediation. So non-human models which have been used is bacteria, prokaryote, yeast, eukaryote, clear, fungus, Cenoherbitant elegans, which is free living, non-pathogenic nematode, invertebrates, Drosophila, fruit fly. This is also invertebrate. And plants. Which plants? Rice, Arabidopsis. This much we have done the sequencing of those organisms which are non-human model to know their natural capabilities so that we can solve the challenges what we face in the agriculture, in the energy production, healthcare or environmental remediation. Now, what are the methods to solve the sequencing? So two approaches were applied. One approach was saying, let us do the sequencing of that part of DNA which is expressed as RNA. So one approach was trying to focus on identifying all the genes which are expressed as RNA. That is express sequence tag. Another approach was saying, so this was first approach, to know only those genes which are expressed. What second approach was saying, let us sequence all. Later on, we will decide which is coding or non-coding area, and that was known as sequence annotation. So two methods were there. One is express sequence tag, in which we are trying to know the sequence which are expressed as RNA. Third was to know the sequence of all. Later on, we can decide coding or non-coding. Now, if you have to do the sequencing, our first step is we have to isolate the DNA. The first step is total DNA is isolated. Then you will convert them into fragments. And after this, we have to clone them in a host by using vector. Because you know what does cloning will result? It will result in the amplification. Now, which are the generally hosts which you use? Because you have to do cloning in the host by using vector. So generally host is bacteria and yeast. And for bacteria, we are using vector that is BAC, and for yeast, we are using vector that is YAC. BAC means bacterial artificial chromosome, YAC means yeast artificial chromosome. Now, how to do the sequencing? So, we are using automatic DNA sequencer, which is based on the principle developed by Sanger. Who is Sanger? Sanger is the one who has done the sequencing of amino acid in protein, and they were arranged based on some overlapping regions present in them. It means you will generate overlapping fragments. And alignment of these sequencing was not humanly possible, so you want special computer-based program. And finally, sequencing was done. So first of all, sequencing target was completed in 2003, but not only they have done the sequencing of 22 autosomes, they have done the sequencing of X and Y. The total 24 chromosomes they have sequenced, and chromosome 1 was completed in May 2006. And this was the last chromosome to be sequenced out of 24. Why we are saying 24? Because 22 autosome and one X and Y separately. Clear. Now, after sequencing, we have to, another challenging task was to know the genetic and physical map of the genes. Then this was generated using information on polymorphism of restriction endonuclease recognition site. This you'll be studying in the biotech chapter. Actually, I'll just give you an idea. Restriction endonuclease is an enzyme which will cut the palindromic site, and it will recognize that palindromic site. So it means that site is known as restriction endonuclease recognition site, or palindromic site, or restriction site. And what we are finding polymorphism in that. Polymorphism is variation at the gene level. So another challenging task was first assigning the genetic and physical map, and this was generated by using information on polymorphism of restriction endonuclease recognition site. So for this, if they give you one word, palindromic site means same only. Because restriction enzyme will recognize the palindromic site. So you have to generate the genetic and physical map by using information on polymorphism on this site. And some repetitive DNA sequence, especially microsatellite. Now, after this, what are the salient feature of human genome? It contained this much million base pair, 3164.7 million base pair. Then total number of genes were 30,000, much lower than the previous estimate. That is early when they started the project, they were thinking genes might be 80,000 to 1,40,000, but how much genes they found? Only 30,000. Then what they found? Function of 50% of the discovered gene they don't know only. It is not known to them. Then less than 2% of genome code for protein. And 99.9% of nucleotide is same because it is making the repeated sequence, which is large portion of the human genome. Now, what are repeated sequence? They are stretch of DNA which are repeated many times, nearly 100 to 1,000 times. Though they don't have any coding function, but they shed light on chromosome structure, dynamics, and evolution. Now, on an average, if you see, we were saying nearly how many genes? 30,000 genes were there. On an average, one gene contained 3,000 bases. But one gene is there, dystrophin, which contained 2.4 million bases. Please learn, average gene contained 3,000 bases and dystrophin contained 2.4 million bases. Chromosome 1 has maximum gene that is 2968 and chromosome Y has minimum gene that is 231. And scientists have identified 1.4 million location where you are finding single nucleotide polymorphism. Means actually change only in one nucleotide. This information will be revolutionary to find the chromosome location for disease associated sequence, and the chances are to know about human history also. So if you have the knowledge from the DNA sequence, then this task, deriving meaningful knowledge from the DNA sequence, will define research through the coming decade, leading to our understanding. This enormous task will require experts and creativity of tens of thousands of scientists from different areas, from public sector, from private sector. And one of the greatest impact of this sequence may well be enabling a radically new approach in the research of the biology. With whole genome sequence and new highlight throughout the technology, we can approach questions systematically and on much broader scale. They can study all the genes, for example, the transcript in particular tissue, why they are going in the organ or in the tumor formation is going on, or tens of thousands of genes and protein work together, how to maintain the chemistry of life. So Human Genome Project, please read very carefully, word to word, line to line is actually very important.